And the species that I've been focused on include the southern toad, which based on previous field studies is thought to have the highest dispersal ability of any of the species. I chose two tree frogs, the green tree frog and squirrel tree frog, which differ somewhat in their habitat preferences, with green tree frogs tending to be more tied to permanent ponds, and squirrel tree frogs basically being extreme habitat generalists. And then the southern leopard frog, which also tends to be tied to permanent bodies of water. And just to give you an idea of the sampling, I ended up collecting all 4 species from almost exactly the same localities, targeting these specific areas across these putative barriers or biogeographic regions. Then to collect the genetic data, I used a target capture approach, the anchored phylogenomics method developed in the London lab where I've been doing my dissertation work. And basically this involves hybridizing DNA probes to capture the same 360 or so loci across all amphibians. I sequenced 36 individuals per species and processed the data using a custom bioinformatics pipeline, lots and lots of manual trimming and Python scripting. Good times. All right, and then to show you the data that I ended up working with, for each species I had over 500,000 sites, around 400 loci, differed somewhat depending on capture efficiency, the number of orthologs per species. Average lengths were about 1,300 base pairs per locus. There were several thousand SNPs within each species. And then last year, I was really excited to learn I could also assemble almost complete mitochondrial genomes just from the raw Illumina sequencing reads. Because mitochondria are so abundant, we just captured them as bycatch. They were just sequenced along with the loci we actually targeted. And so as you might expect, the mitochondrial DNA does a pretty good job of resolving things at the within species level. For the southern toad, there's a lot of well-supported clades, but they don't correspond particularly well with geography. For the green tree frog, the most interesting pattern is that there's this really deep split between just these 2 far west individuals, and using a rough mutation rate from previous studies for the mitochondrial DNA, this mitochondrial divergence is estimated to be over 2 million years old. The squirrel tree frog had a more recent level of divergence, but really strongly supported eastern and western clades. And the leopard frog, again, had a really deep divergence between a western clade and the rest of the range, with a well-supported peninsular Florida clade and Atlantic coast clade. And so the mitochondrial DNA seems to do a pretty good job of resolving things, but what about the nuclear genes? So I estimated an individual gene tree for each locus, and remember they're on average 1,300 base pairs long, but there's still not much resolution in the nuclear gene trees. So this is just summarizing the bootstrap support across all of those gene trees. Average bootstrap support being below 50, most branches not being particularly well supported. But can we still combine all those gene trees to try to estimate a species tree or a population tree? And so I used 2 different summary-based methods, STAR and Astral. And what I found was that for the toad, we have pretty much no resolution within the species tree. There's a well-supported Florida clade, and a couple of those western individuals are popping out. For the squirrel tree frog, same thing, not much resolution. But for the green tree frog and the leopard frog, we find patterns that are pretty consistent with the mitochondrial phylogenies. And so tree-based methods may not be appropriate for within-species studies where we expect there to be high gene flow. So I also extracted all of the SNPs and did a couple of analyses with the SNP data. First off, I was interested in seeing if the strongest genetic boundaries within each species corresponded to those putative barriers or previously described regions. And for the southern toad and the green tree frog, the strongest barrier is just between those 2 far west individuals. For the squirrel tree frog and the leopard frog, there's two boundaries that were detected, but the leopard frog is the only one that has a consistent pattern with one of these previous suture zones. So the western clade and the rest of those clades meet at the Alabama River, which is consistent with the Alabama suture zone. And so finally, to look at the level of genetic structure across species, I used a couple different approaches and found that the southern toad has the weakest structure. The squirrel tree frog has 2 genetic clusters, and then the green tree frog and leopard frog have more structure, and these patterns are really consistent with the species trees and the mitochondrial trees. And so I also wanted to evaluate this hypothesis of expansion from glacial refugia. So I used a spatial diffusion approach in PhyloMapper, which assumes a random walk model of dispersal, to estimate the ancestral center of origin based on a genealogy. And for this approach requires a pretty well-resolved genealogy, so I'm just estimating the mitochondrial genome history here. And for the southern toad, you can see that there's a huge amount of uncertainty. So I'm showing all these dots represent 1,000 random trees drawn from the posterior distribution of trees and the ancestral center estimated for each of those trees. So there's a huge amount of uncertainty there. For the other species, it's a bit cleaner, but one thing to note is that all of these centers are being estimated somewhere in southern Alabama or Georgia, so don't seem to be consistent with that idea of a Florida refugium. And then finally, I applied ecological niche modeling to generate models using MaxCent for both current climate and climate in the last glacial maximum. And I calculated a couple different measures to compare these 2 different climate regimes. So the relative range size is calculated so that negative values indicate range contraction in the last glacial maximum. And for 2 species, the green tree frog and the southern leopard frog, I do find some evidence that there may have been contraction in the last glacial maximum. So the current climate conditions have a much higher suitable area than the last glacial maximum conditions. And then I also calculated an overlap index, so the proportion of overlap between the 2 climate models, where higher values indicate more stable ranges through time. And the southern toad and squirrel tree frog seem to have pretty stable ranges between these 2 drastically different climate models. And if you'll allow me to do a little bit of hand waving, I think one of the most interesting things is that these 2 species that seem to have pretty stable niches are the 2 species that have fairly limited genetic structure and not any evidence of deep genetic divergences. Whereas the 2 species that have these really deep genetic divergences also have limited niche stability and potentially were isolated into separate areas, although probably not in a small Florida refugium as we might have thought previously. But one thing to note is that these 2 data sets are capturing very different time points, so we have to assume that this response to the last glacial maximum climate might have been similar to previous glacial conditions in these species. And that might explain these deep divergences within these 2 species. So I found a lot of discordance across these frogs in the southeastern US. Not much resolution in the gene trees and some of the species trees, suggesting that those first 2 aspects of concordance across nucleotides and loci might not be appropriate. Now that we're collecting these huge nuclear data sets, there's still just not much resolution at these loci. And for these species, there seems to be probably high dispersal in gene flow over time is obscuring the historical signal in these genetic data. But I'd also say that in this particular region, there's probably been a bit of an overemphasis on common barriers, and that the more species we look at, we're probably going to find a lot of discordance. And then one of the things that I think is really interesting to take away is that there seems to be a differential response to historical climate change among these species, even though they all currently occupy this huge area. There's not much evidence for expansion from a Florida refugium, and this idea that the degree of genetic divergence is related to niche stability through time, I think is something that will be really interesting to look at going forward with additional species to try to understand what characteristics actually determine whether species are going to persist through time or be more affected by environmental change. With that, I have lots and lots of people to thank throughout my dissertation, and if there's time, I'm happy to take any questions.
For your species tree analysis, how did you delimit the species?
So for that, I was using every individual as a tip, basically.
Because I mean, I think you get more resolution if you made larger groups. I mean, it's probably not the case that every individual